Today, Homo sapiens is the only surviving human species on Earth. But it hasn't always been this way. 60,000 years ago in Eurasia, we could find various species of Homo, among which Neanderthals, Denisovans, Flores, and Luzonensis. At that time, Homo sapiens had just arrived from Africa and encountered not only environmental conditions different from those to which they were accustomed, but also millennial-scale abrupt climate transitions. Despite this, it managed to colonize Eurasia, already inhabited by other human species. By 40,000 years ago, it continued to inhabit those areas, although with several waves, while the other human species disappeared. What allowed Homo sapiens to thrive under the same environmental challenging conditions? Among the various theories, some researchers hypothesize genocide or epidemics, and others drastic climate changes and food competition associated with exceptional volcanic events. They are all possible explanations for the successful history of modern humans. Venturing off the beaten path, Starch for Sapiens has focused on the role of biological and cultural co-evolution associated with a starch-rich diet. By applying original methodological approaches, Starch for Sapiens integrates three fields of research: archaeology, paleoanthropology, and genomics, aiming to delve into the role played by starchy food for the expansion of Homo sapiens in Eurasia. Our hypothesis is that Homo sapiens, originating from Africa, had an advantage compared to other humans from a diet that included starchy plants, which are nutrient-rich and energy-providing. Starch is found in seeds, roots, and tubers, which are plenty available throughout the year. By contrast, the isotopic data of chemical elements in human bones confirm that the Neanderthals' diet was mainly based on proteins and fats. And this dietary rigidity could have been counterproductive when environmental conditions became harsher once Homo sapiens arrived. Indeed, a complete diet could have had positive effects on Homo sapiens and influenced his demography. Better dietary conditions might have increased the frequency of pregnancies, the quality of breastfeeding, and also reduced maternal and infant mortality. For Homo sapiens, a richer diet could have been responsible for a general extension of life. All of this would have given rise to numerous and collaborative communities, helping in childcare and for the transmission of knowledge. How does the Starch for Sapiens research contribute new data to support the adaptations required for a starch-rich diet? What distinguishes Homo sapiens from Neanderthals and Denisovans in this regard? Let's explore what archaeology can reveal, particularly concerning the functions of pebble stones and the residues still adhering to their surfaces. We have studied stones found during excavations that Homo sapiens used as early as 40,000 years ago to process plants. They were simple river pebbles used to crush and grind tubers, bulbs, roots, shelled fruits, and even seeds. To eliminate possible toxins, plants are typically soaked in running water, then dried, and finally ground to obtain more or less fine flours. They could then be cooked in soups using stones heated in the fire, known as boiling stones. Plants grinding and heat are expected to help the degradation of starch, a type of carbohydrates, during digestion. In Starch for Sapiens, we have found pebbles used for grinding at some Homo sapiens sites dated between 40,000 and 30,000 years ago. And we have analysed them using various technologies: optical microscopes, electron microscopes, SEM, confocal microscopes, even utilising the synchrotron light for chemoprofiling the ancient starch grains. The plant remains retrieved on the stones were rich in starch. We analysed grains with different morphologies and dimensions, and their chemical composition in order to identify them as starch grains. We also managed to understand the function of the pebbles as ground stone tools. Finally, we constructed libraries of starch grains by extracting them from over 150 plants whose species were present in relevant geographical area. Now, let's add the piece provided by paleoanthropology. We have studied with different methods the structure of the bones and the teeth of sapiens and Neanderthals to verify any possible pathologies related to their diet. Sapiens' teeth show various signs of caries, while the Neanderthals' teeth only show 3 caries. The caries and periodontitis observed are caused by well-known bacteria. These bacteria feed on sugar present in starchy food, which are released by the amylase enzyme active in the mouth. Having found so many caries makes us understand that Sapiens' diet was rich in starch. On the other hand, the scarcity of caries in Neanderthals suggests that their diet was poor in starchy food. It was hypothesized that a better nutrition could lead to an increase in fitness and health effects, and our data finally support the notion of a richer nutrition. And finally, what has the genomics taught us? Our study started from fragments of genomes of Sapiens and Neanderthals that we mapped on a genome representative of modern populations. In particular, we were interested in understanding how many copies of the same gene implicated in digestion Neanderthals and Denisovans had compared to archaic Sapiens and modern humans. The hypothesis is that having multiple copies of a gene can facilitate performance of its function. For example, it was observed that, compared to Neanderthals, Sapiens genomes have more copies of salivary amylase genes, the gene that helps us start digesting starchy plants while we chew food. This makes us understand that the Sapiens were, and still are today, particularly efficient in the digestion of carbohydrates, which are the basis of an energetic diet. Expanding the analysis to all the human genes involved in digestion and more than a dozen ancient genomes, we were able to discover new information that integrate that of the amylase gene and give us a more complete picture of the ancient human diet. For example, the Neanderthal genome has multiple copies of genes for digestion of fats, supporting the idea that Neanderthals were genetically better suited to digesting animal fat. Now, let's piece together Starch for Sapiens results from the 3 disciplinary approaches. Through the study of pebbles used as tools, we demonstrated Sapiens knew how to process plants to extract caloric nutrients, specifically starch, from them. The study of human fossils confirms that Sapiens consumed starch-rich foods. The higher frequency of cavities and other gum-related diseases serves as evidence in favor of the significant shift in diet. Through the analysis of variations in gene copies in human genomes over the last 100,000 years, we demonstrate that Homo sapiens was particularly efficient in extracting energy from starch. Hence, cavities were a price to pay to secure a crucial adaptive advantage. The data provided by Starch for Sapiens are consistent with the idea that starch is an integral part of the Sapiens diet well before the advent of agriculture traditionally associated with the Neolithic Revolution over 10,000 years ago. Moreover, the new data contribute transformative idea that adaptation to the starchy diet is the result of a long cultural and biological entangled process. Starch for Sapiens offers new avenues to address the ongoing rapid changes, where technological development and biology are intertwined but follow different paths. By piecing together these elements, Starch for Sapiens contributes a new perspective to establish a connection between human behaviors, migrations, genetics, and adaptations to new environments.